Hello, this is Miss Dagenford again, and I wanted to tell you a little bit about bacteria today. So, our learning goals today will be to understand the structures of bacteria, to understand their shapes, their organization, their oxygen requirements. We're going to do a virtual lab which examines that. And then we're going to end by talking about how we can protect against some of the pathogenic bacteria. So let's start with bacterial cell structures. Now, you had this picture in your homework, so this is a nice review for your homework. So let's start with the cell wall. Now, the cell wall would be this dark brownish red layer, and the cell wall is very protective for bacteria. Now, all bacteria have cell walls, but some have thicker cell walls than others. Now, next to that would be the capsule or slime layer. Some bacteria, remember our R and S cells way back in December. The R and the S cells refer to whether or not they had a capsule, and it also is protective. It means that our immune system does not do a really good job with the ones that have a capsule. It is a cell, so it has a cell membrane as well, and the cell membrane surrounds the cytoplasm and contains it. If we continue to go in, we see, of course, the cytoplasm, which contains all the proteins and sugars and enzymes that the cell needs. If we dive further, we see the main circular chromosome. In this case, it's all kind of wrapped up, but if we were to pull it out, it would be a single loop, just one large chromosome. But a lot of bacteria have these small circular loops called plasmids, and we'll talk about those later. But they're just small small circular pieces of DNA, often containing genes for antibiotic resistance. Often bacteria absorb food from the outside, and so you'll see little food granules like starch or little bundles of protein. They are cells, so they have ribosomes, which make proteins for them. We see these little cell extensions. They kind of look like hair. It isn't hair. It's called pili, and pili are going to be important in helping the bacteria stick to surfaces as well as through a form of reproduction called conjugation. And then we are almost done. We have the bacterial flagella, which basically helps the bacteria to move. Not all bacteria have flagella, but many of them have some. Some have one, some have two, some have a pair on each side. There's just a variation. But that would help the bacteria to move. You're going to watch a series of short films on archaea. And remember, archaea is a type of prokaryotic organism that would be found in extreme conditions. So think about the anaerobic conditions inside a cow's gut or a human's intestine. Think about sewer treatments or swamps where we see very little oxygen, or areas that have a lot of salt or high temperatures. All of these are examples of archaea. These are extreme prokaryotes. We used to kind of just use the term bacteria loosely. They have similar structures to bacteria, so honestly, they would look very similar. But we know now that they have very different genes and very different proteins. One of the most beautiful examples would be the Grand Prismatic Hot Spring in Yellowstone. All of these different colors represent different species of archaea that are existing and growing in different temperature layers in this hot spring. The dark blue would be the hottest, and where we don't see yet any thermophiles. So very beautiful. If you ever have a chance to go out to Yellowstone, absolutely go. It's a beautiful area, very interesting. And so you can see there's lots of different examples here of very salty lakes, in termites, in cows. We have archaea. But the one that probably most students find would be the bacteria that are just living in and around us. So these would be areas that have more typical ranges of living conditions. They have opportunity for food. They need food for energy. They are cells. They can reproduce on their own. They would require a pretty neutral, although some do like weak acidity conditions, which lets us preserve food by pickling. Pickling with vinegar will kill most bacteria, and that's before we had refrigeration and freezers. That was the way that most of our vegetables and even meats to some degree were pickled, and that allowed them to be preserved throughout winter. Thyme is something that we use also in terms of food safety, because bacteria will, with optimal conditions, can divide every 20 to 30 minutes. And that does not take long for you to have a pretty healthy bacterial population. These bacteria live in pretty standard temperature requirements, so they die past like 140 degrees, and that would be pasteurization. So most bacteria are going to live in pretty standard 70 to 104 degree Fahrenheit, typical weather that we have in spring and summer in your house, right? And a lot of bacteria need oxygen, and therefore, when we can foods, we're removing the oxygen. We need a vacuum seal for canning. Again, before refrigeration and freezing, that's where we preserved a lot of vegetables. Now there are exceptions. There are some anaerobic Clostridium, and I spelled that incorrectly, but Clostridium bacteria are perfectly fine without oxygen, and that can be a danger in canning. And so we'll do a virtual lab today that explores that. And then moisture, bacteria need water. So if we remove that, dried foods can preserve that. So bacteria can divide as long as the conditions are fine. Bacteria can divide every 20 to 30 minutes if conditions are optimal. And when they divide, it's very, very simple. It's certainly not like mitosis or meiosis, very simple. Basically, they replicate that main central chromosome, and then the cytoplasm gets bigger, the cell wall gets bigger, and then they divide that in half. That's it. So you can see this very short little video. Let me go ahead and play it for you.
Most bacteria reproduce by a process called binary fission. During binary fission, the parental cell divides to form two daughter cells. In the first step of binary fission, the bacterium replicates or copies its chromosome and attaches each identical copy to a separate location on the cell membrane. Unlike eukaryotic cells, bacteria do not need to dissolve a nuclear membrane or assemble a mitotic spindle, making binary fission very quick and efficient. Second, the cell elongates. Since the chromosomes are attached to different locations on the cell membrane, they separate from one another. Third, the bacterium pinches off at the center and forms a septum, a separating wall between the two compartments of the cell. Finally, the cell splits into two new daughter cells. These daughter cells are fully mature bacteria that can grow and, if conditions are right, continue the binary fission process.
Okay. And you can see in this GIF on the right-hand side, this is a GIF of bacteria dividing. Obviously, it's time-lapse, But you can think about the math involved every 30 minutes. Let's do 30 minutes, it's nicer math. So every hour you have two doublings, right? So it wouldn't be hard to go from 1 to 2 to 4 to 8 to 16 to 32 to 64 to 128 to 250, and so that's one of the things we worry about with food preparation. You can't keep food out in temperatures that allow bacteria to grow very fast, and thus refrigeration. Now, bacteria can be classified by their shape and organization, and so follow along. I'm going to drag these to the right spot. So, bacillus would be rod shape, and so a lot of bacteria are named for their shape. So, for instance, bacillus anthrax would be one, and then we've got the cocci, streptococcus, staphylococcus. And then we have spirilla, which would be, of course, spiral shaped. Those are the three main classifications. Notice some of them are stained pink, some of them are stained purple. We'll come back to that in just a second. And then we have bacteria classified by their organization. So strepto would be in chains. So I'm just going to move some of these down. Sorry about that. I'm just going to move some of these down so you can see them. So strepto would be in chains, and you can make your own strepto. Now, why do we worry about this? Well, this allows us to identify. So if you've ever been to the doctor's office where they do a throat culture, that throat culture allows them to identify visually. So another type of organization would be staphylo. And staphylo would be meaning that they're in clusters. And so I'm going to just copy a couple more. And that just means that if they saw it under the microscope, they would know that that's a staphylococcus, not streptococcus. And then diplo means we have a pair. And so if we saw these guys kind of linking up in pairs, that would tell us what they are. And then just to finish up with this, we've got bacteria classified by their oxygen requirements. Now, notice in all of these tubes, the top of the tube means that there is more oxygen. The bottom of the tube means there's less. So in obligate aerobes, you absolutely require oxygen. You're going to see most of them growing at the top. Sometimes you can see just fewer bacteria down below, but most of them are going to be at the top. If it's anaerobic, it's going to be at the bottom when there's lower oxygen. That's the only place they're going to be able to survive. And then facultative anaerobes, well, they can grow anywhere. They're going to be growing all throughout that. So they can live with or without oxygen. Now you're going to do a lab next, and the lab will be a video that you watch.